Introduction. Welcome to today's lecture on DNA repair mechanisms. DNA is constantly exposed to various forms of damage, including chemical modifications, spontaneous mutations, and physical breaks. To maintain genomic stability and prevent the accumulation of mutations, cells have evolved intricate repair pathways that correct these damages. In this lecture, we will explore 12 major DNA repair mechanisms, ranging from homologous recombination to genome maintenance during DNA replication. Let's dive into the intricate world of DNA repair and discover how cells safeguard their genetic material. Homologous recombination, HR, is a DNA repair pathway that fixes double-strand breaks. It starts with processing the breaks, then invading an intact DNA molecule. Proteins stabilize the structure, allowing for the exchange of DNA strands and preserving genomic stability. HR is important for large and complex breaks and in meiosis. Deficiencies can lead to genomic instability and developmental disorders. Non-homologous end joining is a DNA repair process used to fix breaks in DNA strands. It works by directly joining the broken ends together without requiring similar sequences. This process involves several steps, including the recruitment of specific proteins and the alignment and processing of the DNA ends. Errors can occur during this process, leading to small changes at the repair site. Despite these potential errors, non-homologous end joining is an efficient way to ensure the survival and genetic stability of cells. DNA mismatch repair corrects errors during replication and chemical modifications. MUTS alpha and MUTL alpha complexes recognize and bind to mismatched bases, activating exonucleases and DNA polymerases to remove and resynthesize the mispaired DNA strand. Defects in MMR genes are linked to hereditary cancers. Base excision repair, BER, is an important pathway for fixing damaged DNA. It repairs damage caused by oxidative stress, alkylating agents, and spontaneous hydrolysis. BER involves specific enzymes that recognize and remove damaged bases, cleave the DNA strand, fill the gap with new nucleotides, and seal the break. This process helps prevent mutations, cancer, and neurological disorders. Nucleotide excision repair, NER, is a DNA repair pathway that detects and removes different types of DNA damage. It consists of two sub-pathways, global genome NER, GG-NER, and transcription-coupled NER, TC-NER. The process involves recognizing and binding damaged DNA, making incisions to remove the damaged section, and then filling and sealing the gap. NER is important for maintaining genomic stability and preventing certain diseases like xeroderma pigmentosum, XP. Translesion DNA synthesis is a repair pathway activated when DNA replication encounters damage that slows replication. Specialized DNA polymerases called Y-family polymerases enable synthesis to continue past lesions, though with lower accuracy. TLS is important for preventing replication arrest and has implications for cancer and chemotherapy. Double-strand breaks are severe DNA damage. Two main repair pathways, homologous recombination, HR, and non-homologous end joining, NHEJ, are utilized. Alternative end joining, ALTAGE, and single-strand annealing, SSA, provide backup repair options. These mechanisms protect the genome from harmful mutations and cancer development. Rec helicases are a group of DNA helicases that help maintain genome stability. BLM, a well-known member, is linked to Bloom syndrome and plays a role in DNA repair and replication. Dysfunction of rec helicases can lead to genomic instability and increased cancer risk. Understanding their functions is crucial for understanding human diseases and developing treatments. Fanconi anemia is a rare genetic disorder causing bone marrow failure, abnormalities, and cancer susceptibility. The FA signaling and repair pathway repairs harmful DNA crosslinks by recruiting proteins to incise and restore the DNA through homologous recombination. This pathway helps us understand DNA repair and genomic stability. Cancer involves uncontrolled growth and unstable genes. Problems in DNA repair and signaling contribute to cancer. Dysfunctions in these processes lead to DNA damage and instability, increasing the risk of mutations and tumor development. BRCA1/2 mutations affect cancer susceptibility and response to treatment. Understanding these mechanisms is important for targeted therapies and biomarker identification. DNA repair is impeded by the structure of chromatin. Chromatin remodeling complexes enable nucleosomes to move and reposition, making it easier for repair proteins to access damaged DNA. Histone modifiers and chaperones also play a role in regulating repair. Disruption of this process can lead to disease. DNA replication is a complex process that requires accurate copying of the entire genome. Mistakes during replication can cause mutations and instability. Cells have mechanisms to recognize and fix these errors. Replication stress, caused by factors like DNA damage and conflicts with transcription machinery, poses a challenge. Cells use DNA damage tolerance and replication restart pathways to overcome these obstacles. Proteins like DNA polymerases and helicases coordinate these processes. Proper replication ensures accurate transmission of genetic information, preventing harmful mutations. In conclusion, DNA repair mechanisms are crucial for maintaining genomic integrity and preventing genetic diseases like cancer. 12 major repair pathways, like recombination and mismatch repair, have specific functions and utilize different proteins and enzymes to fix DNA damage. Understanding these repair mechanisms helps us develop targeted therapies and gain insights into diseases caused by repair defects. Ongoing research will uncover more intricacies and provide new treatment options.